Hello, everyone. My name is Alina, and I am in Nanotechnology Engineering. The other members on this project are Terese, who is also in Nanotechnology Engineering, and Matthew, who is in Chemical Engineering. And we will be discussing the new 1B Nano Design Days project, which will be running the first time this upcoming winter term. It is led by Jenn Coggan and Professor Ghavami. So, nanotechnology engineering is a relatively new domain, and the nanotechnology industry is just beginning to grow. With this 1B Nano Design Days project, we have taken an industrial application and are currently developing the science and instrumentation around it. This allows us to create an activity for the nano students which will expose them to various aspects of the program, such as the chemistry side, the electronic side, the software side, and much, much more. The project we have created is nanobio-based, which is appealing to most first-year students as this is what they join the program for. The project is also based on a human-centered problem, which is diabetes, and the successful development of this project would be a step towards some groundbreaking future work. The project in particular is a colorimetric sensor for glucose detection using gold nanoparticles. As myself and the other people working on this project go through the presentation, we will be discussing the four main components of this project, which is nanoparticle optimization, glucose detection with an LFA, imaging, and image analysis. So we began this project with nanoparticle optimization, which involves choosing the right nanoparticles for the colorimetric sensor. Overall, the chemistry portion of this project, and in particular glucose detection with an LFA, has never been explored, and so there was no literature for us to go by. But what we first began with is performing glucose detection in solutions with different nanoparticles. So we first started off with glucose detection on silver nanoparticles, but came to the conclusion that silver nanoparticles are not viable with the LFA format due to the faint color it produces. So as you can see in the picture below, the color it produces ranges from yellow to colorless, which would not be visible on paper. We then performed glucose detection on gold nanoparticles and decided that it is viable for the LFA format due to the distinct color it produces between the different glucose concentrations. So as you can see in the picture below, glucose detection on gold nanoparticles produce this blue color, and a pink color, and then when dropped on a sample pad, you can see in the picture that the colors are apparent.
So there are a few very important reactions that lead to this color change, and we've broken this up into 3 steps. First, the oxidization of glucose forms 2 very important products, gluconic acid and hydrogen peroxide. Hydrogen peroxide is then used to reduce this acid to form elemental gold, AU0. And elemental gold is then deposited onto the gold nanoparticles and causes a growth in size, which is what allows us to see a color change. The dependence on glucose throughout all of these steps is what allows us to see a gradient of colors. So as you can see here at the bottom, the sample has no glucose, and as we move up, increasing in glucose concentration, each sample has a slightly different color. So we are now attempting to move all of those reactions onto paper. And this has involved a lot of research and reaching out to professionals in the field of lateral flow assays for a few reasons. One, because we were formatting it very differently from conventional LFAs. And 2, because we are not only trying to achieve a yes or no answer as to whether our target analyte is present in the sample, but also a gradient of color change that will eventually tell us the concentration of glucose present.
So as Terese and Alina have described, the lateral flow strips will produce a variety of shades of blue, purple, and pink. However, even slight variations in lighting or camera quality can make the same color appear slightly different from test to test. Thus, the strip should always be viewed under controlled conditions. To achieve this, students will be responsible for developing a SolidWorks model of an image box. This design must eliminate as much external light as possible and utilize different configurations of LEDs, LED arrays, and breadboards in order to have a consistent source of internal lighting within the box. This design must also include a mechanism to support the placement of the actual nitrocellulose strip, as well as be compatible with the Orbeck Astra 3D camera, which is the camera we have chosen to use in this project. On the right, you can see an example of one of the image boxes that we have created in SolidWorks, as well as a working 3D model that has been printed and set up with LEDs. Now, once a proper image has been taken, it must be analyzed in order to determine the initial concentration of glucose. Given that the various colors produced are very similar, analysis by the human eye is unreliable, as the same shade of purple, for example, may be interpreted differently by different people. Thus, the images will be run through calibrated MATLAB algorithms that will return the correct glucose concentration of the original sample. Students will be responsible for developing their own functions, and thus will have the opportunity to further develop their MATLAB knowledge that they have begun building during their first year at university.